Also, studies on the repressor protein in the 80s and 90s clarified the structure of the aporepressor, the trypophan activated repressor, and the repressor operator complex. This is the work of Paul Siegler, who also became a tennis pro and a tennis friend, and Oleg Jardetsky, who's now at Stanford. And the repressor is a dimer of a protein polypeptide with a helix term helix. When tryptophan binds, it displaces one of these helices, so the repressor is in a position to interact with 4 base pairs in DNA and bind to DNA. Now, in 1980s, I was convinced or argued with and whatnot by David Perkins to renew my studies with neurospora. At this point, we weren't doing anything with neurospora. And David Perkins used to come, we were always on the same floor, he always used to come to my office and I'd talk with him, and he would complain that only people working in neurospora are geneticists. Nobody's doing any biochemical work at all. And so he wanted me to do some biochemistry, no procedures for cloning in neurospora and so on. So I was convinced and I decided to turn over half of the new graduate student postdocs that came to the lab to neurospora research. But looking back, I'd say that my major contribution in neurospora research was that I trained subsequent leaders in neurospora research. Eric Selker, Dan Eberle, Mike Plemon, Matt Sachs, Mark Orbach, Odette Arden, and Luis Corciano, who gave a seminar here just a couple weeks ago, are all leaders in neurospora research. So I did do some good. But I was mostly interested in the bacterial research. And at this point, I thought we knew enough about regulation in E. coli to begin to look at some other organisms. And Bacillus subtilis was the second most widely studied organism, so I decided to look at this. And I assigned the graduate student, Mitzi Crowder, this project. And we knew at the time that Dennis Henner was sequencing and studying the trypophan. He was at Genentech, and so we collaborated with Dennis Henner in these early studies. And it was known at the time that the trypophan is a suboperon within an aromatic supraoperon with 3 genes upstream, 3 genes downstream, involved in the common aromatic pathway or in allowing tyrosine histidine biosynthesis. One of the TRP genes was not in the suboperon, it's in the folate operon, and Irving Crawford provided an answer for that, and that is that this polypeptide participated in 2 enzyme complexes, and it formed the same reaction in those 2 enzyme complexes, one in tryptophan formation, and the second in folate biosynthesis. So in this organism, you have a single gene producing a single polypeptide that functions in 2 enzyme complexes. That obviously required additional study. So we were, from our inspection of the sequence, it was clear that we were dealing with attenuation. And Paul Golick and Paul Babitsky were postdocs, next postdocs in my lab, were given this assignment, and they studied this. And this predicted anti-terminator and terminator that would form before the first gene. And the way these structures are drawn, this AC3 are the same AC3 that it requires to form the terminator. So therefore, if they are paired in formation of this anti-terminator, they're not available for the formation of a terminator. And the reason I have these triplets, GAG, UAG, marked is that we discovered that the regulation was an RNA binding protein, which I named TRAP, which is a tryptophan activated RNA binding protein that binds to these UAG, GAG repeats, and it melts the anti-terminator, but note there are no GAGs on this side. So when this protein binds, this is free to form a terminator. And eventually, the three-dimensional structure of the repressor-operated complex was solved by Paul Golnick after he left my lab, collaborating with Fred Anson in Britain, who had a crystallography lab. And this shows the structure of the trap protein with an RNA wrapped around it. And trap turned out to be has 11 identical subunits with a tryptophan binding site, tryptophan bound between each adjacent subunit. When tryptophan bound, it alters the conformation of the surface, so the AG of GAG is bound. Okay, so that explains how trap works. So, in the presence of excess tryptophan, bound active trap prevents the formation of the anti-terminator, shown here. The terminator forms, and transcription is terminated. That's in the presence of excess tryptophan. In the presence of excess tryptophan, a new feature was discovered by Babitsky's loop, and that is there's a 5' stem loop before the anti-terminator sequence. And we didn't know what that does, but that 5' stem loop also binds the trap protein, and we think that the 5' stem loop binds tryptophan activated trap, preparing it for access to newly synthesized GAGs in the leader transcript. And we think the possible explanation for only 120 molecules of trap per cell, and the 5' stem loop would preposition trap in position so that it can act when these GAGs are synthesized. Now, in the presence of excess tryptophan, leader termination in the trypophan bacillocellulose is only 90% efficient. What do you do about that? Well, bacillocellulose forms a second RNA hairpin in the unterminated transcript, blocking the initiation of TRPE translation. This was shown by Babitsky, and synthesis of the TRPE protein is further reduced. Again, showing the sophistication of gene regulation. Now, upon tryptophan starvation, trap is trip-free, it cannot bind RNA. The anti-terminator forms and persist. The terminator cannot form, and RNA polymerase continues transcription. What about TRPG? This was solved recently by Babitsky. Babitsky examined TRPG regulation, and he found that the two promoters responsible for production of TRPG and the transcription initiated the second promoter made an RNA which had a trap binding site, which we knew, and when trap binds at the site, it blocks the Charley-Yanofsky region and prevents TRPG translation. So, whenever you make this message, if trap is active, no TRPG protein is produced. How do you deal with the PABA requirement? Turns out that the ribosome translating the PAB messenger RNA displaces trap from the trap binding site, and the PABA protein is synthesized. So there's a very clever way of having one gene specifying two proteins involved in two pathways, but also regulated both by PAB and also by tryptophan. Now this shows the sequence that the trap protein recognizes. It hasn't been, actually hasn't been experiment verified, but you see there's a series of GAGs, UAGs overlapping the Charley-Yanofsky region, and if the RNA is wrapped around the trap protein, the Charley-Yanofsky would not be available for translation initiation. Now, based on our studies with E. coli, of course, the question was raised, is the trypophan of bacillocellulose regulated by trans-RNA, tryptophan trans-RNA, as well as it is in E. coli? And there had been a result in 1974 by Steinberg showing that it isolated temperature-sensitive tryptophan tRNA synthetase mutants so that the enzyme was inactivated at high temperature, couldn't charge tRNA trip. Lo and behold, enzyme expression of the trypophan was elevated. how could that be? We knew there was crap in the cell, trypophan in the cell. Why should that be?